Hey, it's time to start today's UNIST Distinguished Seminar series. Okay, my name is Jongnam Park, and thank you for joining this webinar. Okay. Today, we have a very special speaker in this distinguished seminar, Professor Daeyeon Lee, working in Department of Chemical and Biomolecular Engineering at the University of Pennsylvania. Let me introduce his career. He received his BS in Chemical Engineering at Seoul National University, and he moved to USA and got a PhD at MIT. After finishing his postdoctoral career at Harvard University under the guidance of Professor David Weitze, he joined the UPenn as faculty. So his research focused on the developing deep understanding of interaction between soft materials near or at interface, and extending the obtained knowledge to direct the assembly of microscopic structure with the designed property and functionality. He has won numerous awards, including the 2010 Victor Lama Award, NSF Career Award, 2013 3M Non-Tenured Faculty Award, AICHE NSCF Young Investigator Award, and 2017 Soft Matter Lectureship Award, and so on. He also published more than 200 papers and presented more than 115 times as invited speaker in the department seminar or conference. It's quite amazing for me. Yeah. So let's give a warm welcome to Professor Lee. Hey, Lee, it's time for you.
Okay, let's see. Can you see my screen?
And yet.
Up here, my computer is. Can you see my screen?
Yeah. Okay. Okay.
All right. Very good. So good morning. I'd like to thank. Give a lot of thoughts to the topic that I would discuss to you today, and decided to discuss a thrust in my group that has been developing over who you see on here on the right in bioengineering department. In particular, this talk will feature and highlight our efforts to translate some of the successes that we have had in our laboratories to the industry. It's a topic that I have not had many chances to discuss because it's highly technology-oriented research. But I feel that this presentation hopefully will be something that students can relate to. As a student, I myself thought a lot about what I'm studying, how what I'm studying can impact others. And I know that there are several students that are potentially interested in continuing to work on their own inventions and technologies so that such technologies can be brought to the market and hopefully impact others' lives. So in this talk, I'll focus on our efforts to scale up microfluidic droplet generation for particle manufacturing. I'll briefly discuss why this is important and some of the challenges associated with this work. Of course, none of this work would be possible without the people that are working on it. I especially would like to acknowledge these three gentlemen here, Sagar, Alex, and Hun Ho. So let me, okay, here we go. I would like to motivate this talk by sharing a piece of information that I actually found quite surprising when I heard it first, which was several years ago. But I don't think this is a surprise to any of us now because we are just living through the pandemic right now. So it is uncommon, it is not uncommon that there is a global shortage of injectable drugs and vaccines. And we know this, right? Because we lived through the shortage of mRNA vaccine. And this shortage of these types of injectable drugs or vaccines these problems are not limited. I see something on the chat, so let me see. Oh, you cannot see my screen? Okay. So let me try to stop sharing and let me share again. Okay, that's the case. Okay, let me try to share again. Can you see my screen right now? All right, so it looks like I'm having some technology issues here. So let me try to share my screen one more time. Can you see my screen? Could somebody let me know? Okay, let's see. Yes, very good. Okay, okay. All right. So yeah, let me restart here. So shortage of drugs and vaccines are not problems that are limited to certain parts of the world. It impacts everybody. So here's a study, drug shortage issues related to hepatitis B in the UK. There's also drug shortage in the US, and we just lived through the shortage of mRNA vaccine. And the drug shortage is a serious problem because it's directly related to increase in mortality. All right. So the question is, why is there such drug shortage? Okay. And I thought the drug shortage is because there's shortage on the raw material, but that's actually not true. It turns out that most of the reasons, the most important reasons behind these drug shortages is because of manufacturing issues. Okay. There's some quality manufacturing issues, and also there could be some quality delays and capacity problems. Okay, this means that even if you manufacture some drugs, they cannot be used because there's some safety issues. And one important issue here is that drug industry still uses batch processes. Okay, and this causes a problem because there could be variation from batch to batch. In fact, pharmaceutical manufacturing lags decades behind traditional chemical manufacturing, which has gone to continuous processes. So there's a strong push to adopt these types of continuous processing to produce and manufacture drugs and vaccines. In many cases, when drugs are produced, they're produced based on emulsion-based manufacturing processes. So emulsion, as you know, is a mixture of two immiscible fluids, such as oil and water. When you mix them, you get an emulsion, such as oil in water or water in oil emulsions. These emulsions can then be processed into particles, which can be used as injectables as well as vaccines. Now, traditional methods of creating these particles using emulsions end up giving particles that are extremely heterogeneous, as you can see in this electron micrograph. So we and others have been working on making drug particles that are highly, highly uniform and homogeneous. And we get our inspiration from this right here. This is a leaky faucet. What you see here is that every time a droplet grows, okay, at some point, it will detach from this tap. And the reason for that is that as the droplet grows, it gains weight. And when it gains sufficient weight, gravity pulls it down. And this intricate balance between the gravitational force and surface tension leads to droplets that are always of the same size. And we do exactly the same thing, but at smaller scale. So we create these channels that have dimensions of some millimeter. They're typically tens of micron. And we can now create these droplets one at a time. So here what you see is this is a tapered capillary sitting in another capillary. We have oil that is flowing through this gray area and water that flows around this gray capillary. And the oil gets detached and broken up into these droplets. Okay. So what's really unique about this droplet microfluidics is because you can control the flow of fluids precisely, you can create droplets of exact same size and also different morphologies. So people have been able to make these Janus type droplets. Okay, I'm having some computer issue here again. So let me see if this is going to be okay. I'm very sorry about the computer issues that I'm having here. Okay. Okay, I hope you can still see my screen. So again, just going back to the droplet microfluidics, this droplet microfluidics gives tremendous control over the over the size as well as morphology of droplets. And the field has really taken off. So, these days you'll find, tens of papers that are published every week on using droplet microfluidics to create beautiful droplets that gets used as templates for particle synthesis. Now, there's a huge problem though. The production rate in microfluidics is extremely low. If you're doing 1 milliliter per hour, you're doing pretty good, okay? Whereas in industry, you typically need much, much larger scale. So if you are producing droplets at 1 milliliter per hour scale, to get 100 liters, you need 11 years. And no industry is going to use a technology that does not have the scale-up capability. So while the droplet microfluidics gives you the high uniformity and precision, it lacks the production rate that is required by the industry. So there's this gap between what is being studied in the laboratory and what the industry needs. And this is exactly what I wanted to address. Now, one simple question is, if the flow rate is so low, why don't you just increase the flow rate, right, to get to a higher production rate? And this is the problem, okay? So if you operate at high flow rate, what happens is that you undergo this instability and you induce jetting of the stream. So instead of creating these beautiful droplets one at a time, you induce jetting. And by induced jetting, you no longer have the capability to create these highly uniform droplets. While I won't go into the detail, the reason that increasing the flow rate induces jetting is because the shear force essentially overcomes the surface tension force, or inertial force of this dispersed phase overcomes the surface tension, leading to this jetting phenomena. So simply increasing the flow rate is not going to solve this problem. So one simple way to scale up is to have many droplet generators work in parallel at the same time, right? So instead of having just one droplet generator, you can have 12, you can have 100, you can have 1,000 droplet generators. Problem with that is that each droplet generator will need some kind of liquid handling units, such as pumps. So if you have 1,000 droplet generators, you're going to need 2,000 pumps. And that's not a very viable way to enable scale up. Okay, so but that's what a lot of companies are doing right now. There are actually several companies in this area that are trying to scale up microfluidic technology. The issue with this again is that because you're just scaling up by increasing the number of droplet generator, you need equal number of liquid handling units, and there's variability from droplet generator to droplet generator. Okay, so we thought about this problem a lot, and we actually are very inspired by what happened in microelectronics industry. Okay, so Korea is a very powerful nation in this domain, right? Microelectronics is nothing but taking transistors. Okay, so this is a transistor that was actually first developed at Bell Labs, 1940s. Okay, and to go from here to these semiconductor chips that we have in all of our phones and computers, what needs to happen is that number one, these transistors need to be miniaturized, and then they need to be parallelized onto a single chip. Now, when this parallelization is done, it's done in a way that there's a single channel or conduit circuit that brings in electrons and take out electrons. You don't increase the number of inputs and outputs linearly. So we basically take a similar approach where we have a droplet generator like this, and we parallelize it onto a single chip, and we connect these to delivery channels that will bring in water and oil. And these these droplet generators are all connected in parallel to each other, and they will generate droplets one at a time, and these generated droplets will be collected in this green collection channel. So that's the basic strategy. We are inspired by how microelectronics industry was able to parallelize these transistors to create these semiconductor chips. We want to do exactly the same thing. Instead of controlling the flow of electrons, we are now controlling the flow of liquids. All right, so basically to enable scale up by parallelization, there are several things that we have to consider. And I want to walk through this process step by step with you to show you how we were able to achieve parallelization and what the status of the field is right now. So the first thing that I want to share with you is, how do you design these parallelized devices? So I don't know how many of you work with droplet microfluidics. Maybe none of you do. But there are different ways of generating droplets in microfluidic chips. There are different droplet generator geometries, such as flow focusing, cross flow, step emulsification. Each geometry has its own advantages and disadvantages. What we decided to do is we decided to use this flow focusing geometry because, as you can see in these two plots, where I'm plotting the throughput per device as a function of droplet diameter, or how many drops one can produce per second. Flow focusing device gives the widest range of operation. So it gives the biggest flexibility in terms of producing droplets of different sizes, as well as different throughputs, okay? And then now we have to parallelize droplet generators, okay? And there are two different schemes by which these droplet generators can be parallelized. So I want you to think about this, okay? One strategy is called tree network, okay? So the liquid goes in and then it branches off into two channels, and there are multiple branching points, okay? So it's called tree network. The other geometry that we consider is called ladder network. It looks like a ladder, right? So you've got two fluid streams that are coming in, and then there are these droplet generators that are connected to these two delivery channels. Now, which design do you think offers more advantage in enabling parallelization? Now, the important thing about this is that every single droplet generator has to be fed with the exact same flow rates to be able to produce uniform droplets. So I'm going to tell you that it's actually the right-hand side that we decided to use. The reason for that is the left-hand side is very intuitive, right? Every time branching happens, you divide the fluid into two equal channels. The problem with this tree network design is that if there is a problem with one device, it completely throws off the balance among all the connected droplet generators. Now, then how do you ensure that in this ladder network that every droplet generator is being fed at the same flow rate? It actually becomes a very simple circuitry problem. So like I said, instead of controlling the flow of electrons, we are controlling the flow of fluids. But in the end, they have exact same physics behind them. So let's say this is a delivery channel. So this channel brings in water. And let's say these rungs of the ladder, so these vertical lines, each of them is a droplet generator. We can actually calculate the flow rate through each droplet generator by assigning resistances to different parts of this fluid network. And what you see is that the ratio of the flow rate of Q1 and Q2 can be expressed using this equation right here. And what you see is that it's the ratio of the resistance between the droplet generator in the delivery channel to the resistance of the droplet generator that determines the ratio of the two. And of course, what we want to do is we want to make Q1 as close to Q2 as possible, which means that RD, the resistance between droplet generator, needs to be much, much smaller than RF. And you can do that by making this distribution channel as big as possible. Now, this can be illustrated in this simple figure here. This is a shower stall, okay? And imagine that you're taking a shower over here, okay? If somebody comes in and turns on showers in all the other shower heads, what you don't want to experience is decrease in the flow rate of water through your shower head, right? And one way to ensure that the flow stays uniform across all the shower heads is to make the pipe behind the wall as wide as possible, so that there's very little resistance between the shower head compared to the resistance that is provided by the shower head. All right, so using this approach, what we've been able to do is we've been able to parallelize, in this case, 1,000 droplet generator on a single chip using a single set of inlet streams. So this is water, this is oil. We have two inlets and then one outlet. And these two inlets are now connected in parallel to 1,000 droplet generators using the design rule that I just discussed. So that's how we design these droplet generators and how we parallelize them. Now, how do you make these things, right? So, okay, you can draw these, but as you saw, these designs require three-dimensional fabrication. Your channels need to be arranged in 3 dimensions. And usually the way this is done is that people used to make each layer into an elastomer called PDMS. And then after each layer is formed, these different layers were bonded to each other. But this posed a lot of problems with alignment and so forth. So our group developed this technique where we can actually stamp or pattern PDMS from both sides when it's being cured, so that we can create 3-dimensional monolithic elastomer device as illustrated here. So this device, which is about the size of a US quarter, this is approximately the size of a 500 won coin in Korea. This device has 1,000 droplet generators, and this is what they look like. So this is the flow focusing device that makes the droplet, and this is the big distribution channel that has very low resistance to the fluid. So the water and oil would come through these big delivery channel from outside, and they're delivered to the flow focusing channel using these vias. These things are called vias. And this is what this device looks like when it's under operation. So let me see if I can play this movie. All right, so what you'll see is that these droplet generators, 1,000 of them, are all working in parallel. Okay, and we're moving around in the device to show that they're actually working very nicely. Okay. And if we look at what this looks like, this is the production rate that we see in real time. So using this device, we can create approximately 4 liters of droplets per hour, which is tremendous. All right, so I've talked about the design rule, fabrication. For making these droplets, it's also very important that, and particles, it's important that we use solvents, such as chloroform, dichloromethane. These are common solvents that are used in pharmaceutical industry. Also, it's very important to control the wetting within these devices. Depending on what kind of droplets you want to generate, you may need hydrophobic device versus hydrophilic device. So what we decided to do is, instead of using this polydimethyl siloxane, which is a very, very versatile elastomer to make microfluidic devices, we decided to use this perfluoropolyether polymer. This is essentially liquid Teflon, so it's highly, highly solvent compatible. And as I said, to be able to control the wetting properties of this device, we co-polymerize this liquid Teflon with polyethylene glycol diacrylate, so PEGDA. This PEGDA is very hydrophilic. This liquid Teflon is extremely hydrophobic. So by controlling the ratio of these two polymers, we can control the wetting properties of these elastomers. Okay, so it can go from very hydrophobic to fairly hydrophilic. Okay, and what's great about these devices that you can pump in any kind of organic solvent, and because of the solvent compatibility of this liquid Teflon, these devices don't deform and they operate beautifully. So here what you see is that we can now create water in hexane or hexane water emulsion, depending on whether we have a hydrophobic device or hydrophilic device. And by enabling the production of these droplets using organic solvent, we can make these polymeric particles that have high uniformity. All right, so I've talked about the design rule, as well as how these devices can be fabricated and how wetting and solvent compatibility can be controlled. Now, what I want to talk about is fabrication and solvent compatibility issue again. So what I've shown you is that we can create these devices using these stamping technique, these soft lithography replica molding technique. This is great, but it's not necessarily the best way to produce manufacturing platform. So what we've decided to do is, again, we are very, very motivated by microelectronics industry. As microelectronics industry has done beautifully, we do fabrication directly into silicon wafer. This is a 4-inch silicon wafer, and we now incorporate thousands of droplet generators onto a single chip. And again, using the same design rule that I discussed about 5 minutes ago, we can now have a single set of inlets. So this one is for oil, this one is for water, and these two are the outlets where the generated droplets now exit the device. This, again, takes advantage of the fabrication technology that microelectronics industry has developed. Microelectronics industry can now go down to sub 10 nanometer scale. We don't need that scale, obviously. What we do is we use their technique to create channels that are on the order of tens of micron, which is more than sufficient for us. Now, what we've realized is that in order to create these droplets, what I told you in the beginning is that it's not a good idea to increase the flow rate because you induce this jetting. Okay, so in order to produce, but you obviously want to increase the flow rate, right? So what we found out is that we can create these fluidic resistors right before the droplet generators. So we are decoupling the fluidic resistance by putting these fluidic resistors, and we are decoupling the fluid resistance requirement with the droplet generation requirement. Okay, so by doing this, what we can do is we can actually operate at a fairly high flow rate without inducing jetting, okay, while keeping the resistance of each droplet generator fairly high. So that you can see here. So device 1 does not have any fluidic resistors, whereas device 2 and 3 do have these fluidic resistors. And as you can see, we can maintain this dripping regime where we generate these beautiful droplet one at a time until fairly high flow rate for each device. This means that you can push the droplet generation at high rate for each device. At the same time, you can put many of these devices in parallel onto a single chip. So using this approach, we've been able to incorporate 10,000 droplet generators onto a single 4-inch wafer. And as you can see in these videos, where we are scanning over different regions, we are producing perfectly uniform monodispersed droplets in these devices. And now we've gone up to 20,000 droplet generator, even further optimizing these device geometry. What's great about this lithography technique is that because the lithography is so precise, the droplets are extremely, extremely uniform. In fact, we can't even detect the variability in these droplets, as you can see on this slide. So I just wanted to show you what the droplet generation looks like in real time. So let me see if I can play this movie. You can see that the production rate is extremely, extremely high, and every single droplet that gets produced is exactly the same. So with this setup, now we can easily go above 10 liters per hour production rate, okay, which is tremendous. And this is, in line with what the pharmaceutical industry needs for continuous production and manufacturing of particles for injectables and vaccine. Okay, I'll come back to the question a little bit later. So as I was saying, we have further optimized our device generation by using this trench design. Okay, so we have the big delivery channel that delivers the liquid, right? And then we've got the droplet generators that is sitting on the first floor, and there are these vias. But what we found is that when we try to make these vias, we often end up breaking our devices. So we modified our fabrication technique where we have this little trench dug in, and that helps with integrity of these devices. So our fabrication becomes more reliable by using this approach. So using this approach, like I was saying, we can now pack more than 20,000 droplet generators onto a single chip. All right, so I've covered many different aspects of droplet generation. But in the end, what we want to do is we want to make materials, right? We want to make these particles that can be used in as injectable and so forth. So making droplet is not sufficient. What you often have to do is you have to take those droplets, okay, and then you have to process them so that they become solid particles. Now, the traditional way that people have done this in droplet microfluidics is that they produce these perfect droplets, and then they would collect the droplets and then use bulk emulsion processing techniques to create solid particles. And when you do that, oftentimes what you end up doing is you compromise the uniformity of the droplets, okay? Because you're using, again, bulk method to do the processing. So what we wanted to see is if we can do on-chip processing of the droplets to induce solidification on chip, so that when the fluids come out of the device, we actually have solid particles. So one method that we studied is to solidify these droplets as soon as they're generated in the device on chip using photopolymerization. So the chemistry that we rely on, and in this case, we decided to use hydrogel particles because hydrogel particles, as many of you know, have a lot of utilities. So we take this polyethylene glycol diacrylate. So it's got this polyethylene unit connected to two diacrylates. And we use this photoinitiator called LAT photoinitiator, which has fairly high, fairly fast kinetics. And we induce photopolymerization on chip. What we had to figure out is, what is the residence time of the droplet under the UV to induce solidification, okay? And and this is important to find because we want to make sure that we induce solidification. But obviously, if this becomes too long, that means the footprint of each droplet generator is too big, which will reduce the number of droplet generators that we can paralyze onto a single chip. So we found out that, for this particular formulation, we needed approximately 90 milliseconds or longer time to induce solidification or gelation of this precursor. So again, using the exact same paralyzation technique that I described, we now paralyze these droplet generators, and we then induce photopolymerization right upon droplet generation to induce the gelation. And as you can see, when these gels are cured or polymerized on chip, the hydrogels that we collect retain the original uniformity of the droplets. Whereas if you try to do the curing off chip, you often run into this issue where you have now lost the uniformity that you originally had when the droplets are generated. So this gives tremendous advantage in retaining the uniformity and the precision that microfluidic allows. Now, something that we wondered and realized is that you need to irradiate UV for a certain period. But what if we change the time under which the droplet spends being irradiated by the UV. So in other words, what if we change the time of UV radiation or photo cross-linking? And we can do this simply by changing the flow rate. Okay, and what we wondered is, can we change the mechanical properties of these hydrogel? Okay, because as many of you know, mechanical properties of hydrogels have enormous control, can impact what happens when they're injected or when they're interacting with the cell. So my colleague here, Dennis Disher, has shown that by controlling the stiffness of hydrogel, it is possible to affect the differentiation of stem cells into different lineages. Okay, so our hypothesis was by controlling the irradiation time, we can control the mechanical property. And as I said, studies have shown that the mechanical property of hydrogel is the key factor that determines the fate of cells that are interacting with them. So in order to measure the mechanical properties of hydrogel, what we had to do was we had to come up with a method to characterize their stiffness. And there's this really nice paper that was published by Dave Waits about 10 years ago, where they push hydrogel into this tapered channel. Okay, and by analyzing the shape of these hydrogel, okay, that is being pushed into this taper, and relating that to the pressure that is being exerted, it is possible to estimate both the compressive modulus as well as shear modulus. And as you can see, as we increase the exposure time, again, this is the time under which these droplets spend under UV radiation, the modulus can be controlled from hundreds of pascals to a few kilopascal. And this is the perfect range for hydrogels to be used for different biomedical applications. Okay, so I've covered all these different aspects of scale up. Okay, so where are we right now? I'm very excited about this scale up because now we can really bridge the gap between academic laboratories and the market and the industry. So this is what I call our Morse chart. Okay, so this is sort of different studies that have shown that scale up of droplet microfluidics. And as you can see, we are right here right now. Okay, so we currently hold the world record in terms of number of droplets that can be generated per time, and how many droplet generators can be paralyzed. Now, there's no reason for us to go to even bigger wafers, such as 12-inch wafers, which what Samsung uses to make all the semiconductor chips. The reason that we stopped at 4-inch wafer is because this is the facilities that we have at our university, at the University of Pennsylvania. We are very excited about this development. So along with my colleague Dave Isidore and Saga Yadavali, who used to be our joint postdoc, we've launched a startup, Infinite Fluidics, and this company is actually doing quite well. Okay, so what does this mean right now? We have this 4-inch chip that can generate droplets at tens of liters per hour scale. Is that going to actually, is that something relevant for the industry? So we did a little case study here, and I'd like to share this with you. So there's this anti-retroviral therapy that is being developed, that has been developed for people with that are living with HIV. Okay, and these are injectables. So these are made in forms of particles. Okay. And we basically did a back of envelope calculation to see, how many chips do we need to run in order to meet the world's demand for this particular drug? Because it's a growing market. And what we found is that if we operate 100 of our 10K chips, so each 4-inch chip has 10,000 droplet generators, and if we operate 100 of these, we can meet the world demand for this particular drug. And it's not inconceivable that hundreds of 100 chips are being operated simultaneously at a plant that would make these types of drugs. So we are already at the scale where industry can take our technology and start manufacturing their drug products. There's another very unique advantage of our paralyzation technique. what you saw is that we can put many these devices where each device is generating exact same droplet. Now, as many of you know, when drugs are developed, they go through different trials and phase studies. So initially you start with preclinical, laboratory scale with, cells and small animals, and then they go through these, different phase studies. We saw this with mRNA vaccine as well. Now, what happens in pharmaceutical industry is that for each phase and stage of development, you have to have a new way of producing drug products, okay? And that incurs a lot of cost and time associated with developing process that will create sufficient amount of materials for each phase of the study. Whereas in our case, we just have to tune the number of droplet generators that would get incorporated into a single chip. So from preclinical trial all the way to the market scale, the basic droplet generation unit is exactly the same. So there's no need to develop a completely new process to create these drug particles. All we have to do is just change the number of droplet generators that are parallelized on a single chip, and a single platform can address the entire process of drug discovery as well as manufacturing, which I think is really exciting. All right, so with that, I might be finishing a tad bit early, but I hope I didn't go too fast. What I've shown you is that by miniaturizing droplet generation to these micro scales, and by using the strategy that was used by microelectronics industry to Paralyze, we can now produce highly uniform droplets from a single chip without increasing the number of liquid handling units that is required to operate these devices. In order to scale up, I've shown you that there's a specific design rule that one has to be aware of, and one has to be able to fabricate these devices in 3 dimensions. Okay, it's also very important to control the wetting, and I've shown you how we can use either silicon-based system or liquid Teflon system to enable use of different types of solvents. And by enabling solidification of these droplets on chip, we can retain the uniformity and precision that microfluidic affords. We are now expanding our scale-up efforts into these more complex emulsion droplets. So in this case, what you see is these compound bubbles where you have gas surrounded by oil dispersed in water. And we are very hopeful that these types of particles can be used in different types of drug pharmaceutical manufacturing. So I hope that I was able to convince you that microfluidics is not just for the lab scale. We can actually take this and use it for manufacturing in pharmaceutical industry. So with that, I'd like to thank again the people who worked on the project. So Saga Yadabali, as I said, was a joint postdoc between myself and Dave. So he's now the CEO of this company, Infinite Fluidics. Hun Ho was, again, another postdoc that we jointly advised. He's now a professor at Jeonnam National University. Alex is a PhD student that's finishing up. And of course, I'd like to thank our funding agencies. I would welcome any questions from the audience. If you have questions, you can ask me in Korean. I speak very good Korean. And again, thank you very much for the invitation. Thank you.
Hey, thank you for your very brilliant talk, Professor Lee. And now our seminar is open to the question and seminar the comment. Okay. So the first question in the chatting room, he Park asked about the dimension issue of your droplet. So
yes. So yeah, Professor Park, thank you very much. So right now, the droplets that we are producing are approximately tens of microns. We can easily go down to a few microns, and we are pretty confident that we can go below some micron if necessary. The reason for that is because we are doing this fabrication in silicon chip, the resolution of the fabrication is extremely high. Now the problem becomes the back pressure. As you decrease the size of the channel, the pressure goes as R to the fourth. So we have to have a strategy to be able to deliver very high pressure if we want to make small particles. But we don't think it's a problem with the silicon-based chips.
So let me ask one quick question. So related to
Sure.
the first question, I think some pressure can be developed in your three-dimensional, the monolithic electron device. So how much the pressure can be your device withstand?
So our silicon and glass chip, it's created by. So I went through that way too fast, I think, for the audience. So I apologize for that. But let me maybe I can go back to the slide where I talk about that. So this device is made by doing most of the actual fabrication in the chip. Okay. And what we do is after the chip is fabricated, it's bonded, anotically bonded to glass, okay? And this bonding is extremely strong, so it will withstand, you know, hundreds of psi. So you know, pressure is not a problem. You know, applying pressure can be a problem. The device itself can withstand, you know, very, very high pressure.
I see.
And we typically operate at around 60 to 70 psi.
Okay. So another question from the chatting room. Manhibyeon. The question may come from ignorance for microdroplet. Could you please explain the system in aspects of energy or economics, such as reduction, increase of the expense in use? So basically the question is, how much is it to fabricate your device?
Yeah. So this is a wonderful question. So the device itself is not that expensive, actually. Okay, it's actually the manufacturing facility that you have to have access to. Now, what's great about this is that microelectronics industry again has created these foundries. Okay, so if you have the device design, you can basically order your devices from these foundries. Okay. I've shown you these companies that are selling different types of products right now in the market. So here, so these companies are charging enormously high prices for these devices, these quote unquote paralyzed devices. But we believe our approach can produce droplets at scale that is much greater than these technologies can, while keeping the cost low, because we don't necessarily need large number of liquid handling units.
And additional question from the Manhibyeon is that how large is your system, right? When you produce the 4 liters per hour product. So actually he asked the dimension of your final device.
Right. Very good. So I actually have. There we go. So the device that was generating at 4 liters per hour, this device is about the size of a quarter, 500won. Okay, so this is about size of 500won coin. The device that had 10,000 or 20,000 droplet generators. Okay, these devices, it's about 4 inches, so it's about 10 centimeters in diameter. It's about the size of your hand.
Okay. And you can also use the raise hand icon, raise hand function. Right? You can directly ask the Professor Lee.
Okay, so here's a question. Thank you for very impressive. Thank you. You moved from PDMS to fluoropolymer and finally silicon glass bonded chip. Their surface chemistry is very different, and I wonder whether you had to optimize surfactant or any operation conditions according to different chip materials. Very good. So what we found is that if there is a formulation that works in one device, they tend to work in other devices as well. Now, what's really important is I've shown you how I can control the wetting properties of fluoropolymer-based device, right? It is also important to control the wetting properties of silicon-based device. And what we do is we use silanes, commonly available silanes, to either make them hydrophilic or hydrophobic. So that is very important. But in terms of the actual formulation, we didn't see much difference between, you know, PDMS device, fluoropolymer, and silicon-based devices.
Okay. So any other question or comment? We have around 10 minutes left. So no more question and okay, so if there is no more question or comment, then we can close our session here. Okay. So thank you for joining this webinar again. And, okay, yep. Thank you, Professor Lee.
Thank you. Thank you for
Yeah.
the invitation again.
Okay. Yeah. Have a good night. Bye.
Thank you. Have a great day.
Yep.